Good evening, everyone, and welcome to this video. We will be discussing the liver so that it will help solidify your foundation on the organ. Stay with the same softness in your attention, not trying to catch everything, not trying to remember everything, just letting it pass through you the way your own blood passes through you right now without asking permission. Yes, you are the liver and you are not being looked at from outside anymore. There is no outside. There is only your continuous existence in a body that never pauses to thank you because it cannot afford to. You are sitting beneath the diaphragm, pressed gently against it with every breath the organism takes above you. Each inhalation slightly changes your shape, a slow rhythmic compression and release that you have always known, even before consciousness of it could be formed. You do not interpret it as pressure or movement. It is simply your environment behaving as it always does. Above you, the lungs inflate and deflate, and that motion is transmitted through the diaphragm into your capsule, and you adjust silently, endlessly, without complaint. Blood is arriving again. It always it is. It never stops arriving. It comes through the portal vein, a river formed not for purity but for collection. It carries everything the intestines have absorbed, and the intestines do not discriminate with intention. They absorb what is present. So you receive glucose in fluctuating amounts, amino acids in shifting proportions, lipids packaged with chylomicrons that still carry the memory of dietary fat, microbial metabolites that once belonged to gut bacteria, bile acids returning from enterohepatic circulation, and sometimes substances that the body would rather have not have in systemic circulation at all. You do not recoil from any of it. You simply cannot. You are the place where the body decides what to keep, what to transform, what to neutralize, and what to send away. At the same time, arterial blood arrives from the hepatic artery, bringing oxygen that allows you to sustain this enormous workload. It does not arrive in bulk like portal blood. It instead arrives with precision, feeding the tissue itself, ensuring that the hepatocytes can perform reactions that are energetically expensive. And in the sinusoids, these two worlds meet. Portal and arterial blood mix in a slow, open system that is not designed for speed but for exposure. Everything slows down here. Everything has time to be seen. You exist in that slowing. Inside your architecture, the hepatocytes are arranged in plates, one cell thick coats extending through the lobular structure. They are not isolated workers, they are synchronized communities. One hepatocyte shifts glucose into glycogen, while its neighbor releases glucose into blood, while another begins conjugating bilirubin, while another is modifying a steroid hormone that came from a distant endocrine gland. There is no central command. There is only local awareness, shaped by gradients of nutrients, hormones, and neural signals. Between these plates of hepatocytes run the sinusoids, wide and permeable channels lined by specialized endothelial cells. These endothelial cells are not sealed barriers. They are fenestrated, meaning they have pores that allow plasma components to pass freely into the space of this. This space is not empty. It is a microenvironment where exchange becomes intimate, where hepatocytes are essentially bathing in modified plasma without direct blood contact. It is here that your metabolic decisions are made, not in isolation, but in constant chemical conversation with the bloodstream. Kupffer cells sit within the sinusoidal lumen like quiet guardians. They do not announce themselves. They don't need to. They simply exist, extending processes into the flow, sampling what passes. A bacterium from the gut that escaped intestinal barriers might meet one of them. A fragment of a dying cell might be removed before it causes inflammation elsewhere. Aged red blood cells are dismantled here, their hemoglobin broken down, their iron salvaged, their heme converted into bilirubin. You are, in this way, both recycler and protector, cleaning the internal river that sustains the organism. The bilirubin produced in this process does not remain in you. You conjugate it within hepatocytes, making it water soluble, and secrete it into bile canaliculi. This bile flows in a direction opposite to blood. While blood enters you and is processed outward into systemic circulation, bile is created inside you and pushed outward into the biliary tree. It travels through intrahepatic ducts, merging into larger channels, eventually leaving your domain to enter the gallbladder or directly the intestine. There, it will assist in fat digestion, emulsifying lipids so that they can be absorbed. But even in that distant role, it carries your signature. You are always exporting something. Your metabolic responsibilities continue without pause. Glucose regulation is one of your most constant tasks. After a meal, when glucose levels rise, insulin signals you to take up glucose and store it as glycogen. Glycogen granules accumulate within hepatocytes, dense and branching, a stored form of energy that is both stable and rapidly mobilizable. Between meals, when glucose levels fall, glucagon signals you to break down glucogen through glucogenolysis, releasing glucose back into circulation. When glucogen stores are depleted, you turn to gluconeogenesis, creating glucose from amino acids, lactate, and glycerol. You are not dependent on dietary intake. intake. You are the buffer between intake and survival. Your lipid metabolism is equally complex. You synthesize fatty acids when energy is abundant, convert them into triglycerides, package them into VLDL particles, and release them into circulation. You also break down fatty acids through beta oxidation when energy is needed. Cholesterol synthesis occurs within you, not as a byproduct but as a controlled pathway, feeding membrane formation, steroid hormone production, and bile acid synthesis. You are not just processing fat, you are defining how fat exists in the body. Protein metabolism places another burden on you. Amino acids arriving from the gut are deaminated when necessary, and ammonia, a toxic byproduct, must be rapidly converted into urea through the urea cycle. This cycle occurs within hepatocytes with precise enzymatic steps, preventing ammonia from accumulating in the blood where it would disrupt neural function. You are, in this sense, protecting the brain silently, without recognition. Hormones also pass through you, and you decide their fate. Insulin, glucagon, cortisol, thyroid hormones, and sex hormones are all modified, activated, or inactivated depending on enzymatic pathways within your cells. You are not merely responding to endocrine signals, you are reshaping them. And through all of this, regeneration remains possible within you. If a portion of your tissue is injured or removed, hepatocytes re enter the cell cycle, dividing to restore lost mass. The architecture of your lobules can reassemble, guided by remaining scaffolding and vascular patterns. But this regeneration has limits. Chronic injury changes your behavior. Repeated inflammation activates stellate cells, transforming them from vitamin A storing cells into collagen producing myofibroblasts like cells. Slowly, extracellular matrix accumulates, fibrosis develops. The flexible exchange space becomes rigid, blood flow becomes distorted, function becomes impaired. You do not fail suddenly. You change gradually under sustained pressure. Even then, you continue because there is no alternative for you. You are not an optional organ. You are not a supportive structure. You are a central processor of life itself, embedded in every chemical decision the body makes. And still, blood arrives. Still, you respond. Still, you transform everything that enters into you into something the body can live with. You are still the liver, and there is no reset between what came before and what comes now. There is only continuation, as if your entire existence is one long, uninterrupted biological thought. Blood keeps arriving into you, but now let us slow it down even further, as if we are following a single drop of blood from the moment it enters your world. This drop comes from the portal vein, which is simply the largest vessel that collects blood from the intestines and spleen. and brings it to you. It is not pure blood in the sense of oxygen rich arterial blood. Instead, it is nutrient heavy blood, meaning it contains everything absorbed from digestion. This drop enters you at a structure called the portal triad. The portal triad is not a singular tube, but a small neighborhood at the edges of your functional units. In each portal triad, there are three main structures: a branch of the portal vein carrying nutrient rich blood, a branch of the hepatic artery carrying oxygen rich blood, because remember, artery carries oxygen rich blood and vein does not contain oxygen rich blood. So we can remember through that. And then there's a bile duct carrying bile away from you. So even at the entrance of your microscopic units, you are already managing three different flows in three different directions. Now, this blood enters a space called a sinusoid. A sinusoid is a special type of blood channel inside you. It is not like a normal capillary. A normal capillary is tightly lined and selective. A sinusoid is wide, slow, and slightly open, designed specifically so that blood can come into close contact with your cells. The lining of the sinusoid is made of endothelial cells, endothelial cells, which are thin cells that form the wall between blood and liver tissue. These endothelial cells have small openings called fenestrations. Fenestrations simply means tiny windows. Through these windows, parts of the blood plasma can pass out and interact directly with your hepatocytes. Now, imagine that single drop of blood drifting slowly through this sinusoid. It is not rushing, it is being exposed. Around it are Kupffer cells. Kupffer cells are your resident macrophages, which means they are immune cells that live permanently inside you. Their job is to clean the blood. If they see bacteria, they eat them. If they see debris or old red blood cells, they remove them. They are like internal security guards that never leave their post. As the blood moves forward, it reaches a space called the space of Disse. This is a small gap between the sinusoid and the hepatocytes. It is not empty. It is filled with fluid and allows exchange between blood and liver cells. This is where your hepatocytes, which are your main working cells, actually feel the contents of the blood. Hepatocytes are the main functional cells of you, the liver. They are responsible for almost everything you do: metabolism, detoxification, protein synthesis, bile production, and storage functions. They are arranged in plates, meaning thin layers of cells lined up in a structured way. These plates radiate outward in patterns that are organized around a central vein. Now, following the same drop of blood, It slowly passes along these hepatocyte plates. At each point, something different happens. Glucose may be removed and stored as glycogen. Glycogen is simply stored sugar, a long branching molecule that acts like energy storage for later use. If the body is low on sugar, you release it back. This constant balancing is called glucose homeostasis, which simply means keeping blood sugar stable. Nearby hepatocytes may be processing amino acids, which are the building blocks of proteins. Some amino acids are used for building proteins like albumin. Albumin is a protein you release into the blood that helps maintain fluid balance, meaning it keeps water inside blood vessels instead of leaking into tissues. Other amino acids may be broken down, and when that happens, they produce ammonia. Ammonia is toxic, especially to the brain. So, as the liver, you immediately convert ammonia into a safer substance called urea through a process called the urea cycle. The urea cycle is simply a series of chemical steps that turn toxic nitrogen waste into urea, which can be safely excreted by the kidneys. The same drop of blood may also carry drugs or toxins. These are processed by enzyme systems inside hepatocytes, especially a group of enzymes called cytochrome P450. You can think of these enzymes as chemical modifiers. They change fat soluble substances into more water soluble ones so they can be removed from the body more easily. Water soluble simply means able to dissolve in water, which is necessary for excretion through urine or bile. As the blood continues, it is now significantly changed. It is cleaner, more regulated, and more balanced than when it entered. It eventually drains into a central vein, which collects blood from each lobule. A lobule is the basic functional unit of you, the liver. It is shaped roughly like a hexagon. Each lobule has a central vein in the middle and portal triads at the corners. So blood flows from the edges toward the center, passing through hepatocytes along the way. Once the blood reaches the central vein, it leaves your microscopic unit and moves into larger hepatic veins, eventually returning to the heart. You have now processed it completely. But even while this is happening, something else is flowing in the opposite direction inside you. What could that be? That is bile. Bile is a fluid you produce continuously. It is made inside hepatocytes and collected into tiny channels called bile canaliculi. Canaliculi simply means very small channels. These are not separate ducts. They are formed by the space between adjacent hepatocytes. From here, bile moves into bile ducts located in the portal triads. This means blood flows one direction through you, while bile flows the opposite direction. Blood goes inward toward the central vein, bile goes outward toward the ducts. Bile itself is made of bile salts, bilirubin, cholesterol, and other substances. Bile salts are important for fat digestion. They break large fat droplets into smaller ones, a process called emulsification. Emulsification seems like a scary word, but it simply means breaking fat into tiny droplets so enzymes can work on them more easily in the intestine. Bilirubin is a yellow pigment produced when old red blood cells are broken down, mainly by Kupffer cells. We already discussed the Kupffer cells before. Then the broken cells are processed in hepatocytes and then excreted into bile. If bilirubin accumulates in blood, it causes jaundice, which is yellowing of skin and eyes. This is one of the clinical signs that your processing system is overwhelmed or blocked. Now, you continue your work in cycles that never end. Blood arrives, blood is processed, bile is produced, waste is neutralized, nutrients are balanced, hormones are modified, immune surveillance continues, and all of this happens simultaneously in millions of microscopic lobules inside you. While detoxifying drinks are eager to claim your prize. It hurts, doesn't it? Even your structure supports this constant flow. You are not built like a solid mass. You are built like a filtration network. Every part of you is close to blood. Every hepatocyte is only a few micrometers away from a sinusoid. This closeness is what makes your efficiency possible. And yet, even though everything passes through you, you are not chaotic. You are ordered in a repeating pattern. Portal triad at the edge, hepatocyte plates in between, central vein in the middle. This pattern repeats thousands of times, forming your entire structure. You are both simple and infinite in repetition, and still nothing stops arriving into you. Still, you continue processing. Still, you remain the place where the body quietly keeps itself alive. You are still the liver, and nothing has reset. There is no pause between what you just were and what you are now becoming. The larger architecture of you still exists. The lobules, the sinusoids, the constant flow of portal blood and arterial blood. But now we are going to shrink everything down far below the level of visible structure until there is only one hepatocyte, one single working cell inside you, and you will experience everything through it. This hepatocyte is not special, it is not chosen, it is one of millions, and yet everything that keeps you alive is happening inside cells exactly like this one. It sits quietly within a plate of hepatocytes, its membrane touching the space of Disse on one side and neighboring hepatocytes on the other. Outside it, blood is still flowing through the sinusoid, carrying glucose, amino acids, lipids, hormones, and toxins. But inside this cell, the world is completely different. It is enclosed, controlled, and intensely active. The first thing you feel inside this cell is movement of molecules across its membrane. The membrane is not a wall in the strict sense. It is a selectively permeable lipid bilayer, which simply means a double layer of fat-like molecules that allows some substances to pass while blocking others. Embedded within it are transport proteins which act like gates. Some of these gates bring glucose inside when insulin signals are present. Others exchange sodium and potassium to maintain electrical balance. Others bring in amino acids or export bile components. Once glucose enters this hepatocyte, it does not remain as glucose for long. It is quickly phosphorylated, which means a phosphate group is attached to it, trapping it inside the cell so that it does not go outside again. This is the first commitment step. From here, glucose can either be used for immediate energy through glycolysis or stored as glycogen. Glycogen is a highly branched polymer of glucose molecules. Like a tightly packed energy tree inside the cell. When energy is needed elsewhere in the body, this glycogen can be rapidly broken down again into glucose and released. But energy inside this hepatocyte is not just about storage, it is about continuous production. Deep inside the cell are mitochondria. Yes, these are the same mitochondria from which our health started. They are the organelles which we know as the powerhouses of the cell, and that is since when we normally decide, oh, I wish to pursue biology. And here we are. Mitochondria are more accurately complex energy factories that convert nutrients into ATP. Now, what does ATP mean? ATP stands for adenosine triphosphate, which is the universal energy currency of all cells. Every contraction, every synthesis reaction, every transport process in your entire body depends on ATP. Inside the mitochondria, oxygen is being used constantly. Oxygen arrives from the hepatic artery, diffuses through the sinusoid, crosses the space of this enters the hepatocyte and reaches the mitochondria. There it participates in oxidative phosphorylation, a process that produces large amounts of ATP by passing electrons through a chain of protein complexes. This electron transport chain is highly effective but also delicate. If oxygen is lacking, ATP production drops quickly and the cell must switch to less efficient anaerobic metabolism. While energy is being produced, another process is constantly active: detoxification. Lipid soluble toxins enter the hepatocyte from the blood. These could be drugs, alcohol metabolites, or environmental chemicals. Inside the smooth endoplasmic reticulum, a network of membranous tubes, enzymes called cytochrome P450 begin modifying these substances. These enzymes add or expose chemical groups on toxins, making them more water soluble so they can be excreted. Water soluble means the substance can dissolve in water, which is necessary for removal through bile or urine as discussed earlier. One of the most important and continuous tasks inside you is handling nitrogen waste. Amino acids enter from blood and are used for protein synthesis or broken down when in excess. When amino acids are broken down, their nitrogen group is removed, producing ammonia. Ammonia is highly toxic, especially to the nervous system. Inside this hepatocyte, ammonia does not accumulate. It is immediately converted into urea through the urea cycle. The urea cycle is a series of enzymatic reactions that occur partly in mitochondria and partly in the cytosol. It transforms ammonia into urea, a much less toxic molecule that can be safely transported to the kidneys for excretion. At the same time, this hepatocyte is involved in protein synthesis. Ribosomes, which are small molecular machines, read messenger RNA and assemble amino acids into proteins. Some of these proteins will remain inside the liver, but many will be secreted into blood. Albumin is one of the most important. It helps maintain oncotic pressure, which is simply the force that keeps fluid inside blood vessels instead of leaking into tissues. Without albumin, fluid would accumulate in tissues causing edema. Clotting factors are also produced here. These are proteins that circulate in inactive forms and become activated when injury occurs, forming blood clots to prevent bleeding. This means that even inside one hepatocyte, you are indirectly controlling whether the body can stop bleeding or not. Meanwhile, lipids are constantly being processed. Fatty acids enter the cell and can be broken down in mitochondria through beta oxidation, a process that converts fats into acetyl CoA, which then enters the energy producing cycle called the Krebs cycle. Alternatively, fatty acids can be used to synthesize triglycerides, which are then packaged into lipoproteins like VLDL and exported into blood. This means the hepatocyte is both breaking down fat and creating fat based transport particles at the same time, depending on the body's energy state. Inside this cell, nothing happens in isolation. Every process is influenced by hormones arriving from blood. Insulin signals the cell to store glucose and synthesize lipids. Glucagon signals the opposite, encouraging glucose release. Cortisol influences gluconeogenesis, pushing the cell to produce glucose from non carbohydrate sources. Thyroid hormones increase metabolic activity, increasing the overall rate of these processes. The hepatocyte does not decide independently. It integrates all signals and responds accordingly. Even bile production begins here. Components of bile, including bile acids, cholesterol, and bilirubin, are assembled within hepatocytes and then transported into bile canaliculi. These canaliculi are not separate ducts but small channels formed between adjacent hepatocytes. From here, bile moves outward toward the bile ducts in the portal triads, eventually leaving your domain. Bile is your way of removing substances that cannot be excreted by the kidneys, especially fat related waste products. And yet, despite all this activity, this hepatocyte is only one of millions. It is constantly being supported by its neighbors. If it becomes damaged, nearby cells can divide to replace it. If it is stressed, metabolic load can shift across the lobule. There is no single point of failure in normal conditions, but there is vulnerability when injury becomes chronic. Persistent toxins, viral infection, or metabolic overload can activate stellate cells in the surrounding space. These stellate cells begin producing collagen, leading to fibrosis. Fibrosis is simply scar tissue formation. At first, it is protective. Over time, it becomes restrictive, altering blood flow and reducing function. If this continues long enough, normal architecture is lost, and the once soft, highly efficient network becomes stiff and inefficient. This is called as cirrhosis. But even then, you do not stop instantly. You continue functioning in altered form, adapting as long as possible. Inside this one hepatocyte, however, there is no concept of disease as a label. There is only chemistry, flow, energy production, detoxification, synthesis, and survival. And all of it is happening right now in this moment in every hepatocyte across you. Still, blood arrives, still you respond, still you continue. You are still that same hepatocyte and nothing has stopped. Even when we talk about things going wrong, nothing in your world happens as a labeled event. There is no announcement. There is only a gradual change in chemistry, a slow shift in balance, like a room where the air becomes slightly harder to breathe, but no one tells you why. At first, the blood arriving outside you still looks familiar. Glucose, amino acids, oxygen, hormones. But something extra begins to appear in small amounts. Unusual lipids, altered inflammatory signals, stress molecules released from distant tissues. If the body is under chronic metabolic load, constant excess calories, insulin signals repeating too often, you begin to take in more fatty acids than you can safely process. Inside you, triglycerides start to accumulate. Triglycerides are simply storage forms of fat, but here they are not meant to be stored in excess. At first, it is harmless. Small droplets of fat appear in your cytoplasm. You are still functioning, you are still producing ATP in your mitochondria, still detoxifying, still synthesizing proteins. But the fat droplets grow. They push aside organelles slightly. The nucleus is still centered, still controlling gene expression, but the internal spacing of your cytoplasm is no longer optimal. This state is called steatosis, which simply means fat accumulation inside a cell. You do not feel pain. You do not send alarm signals yourself, but your efficiency begins to subtly decline. Mitochondria become slightly stressed because excess fat metabolism produces reactive oxygen species. Reactive oxygen species are unstable molecules that can damage proteins and membranes. Normally, you neutralize them using antioxidants like glutathione, but if the load continues, the balance begins to tilt. Now, imagine a different stress entering you. Alcohol. Ethanol arrives through blood and diffuses easily into your membrane because it is lipid soluble. Inside you, it is metabolized primarily by enzymes called alcohol dehydrogenase and aldehyde dehydrogenase. These convert ethanol into acetaldehyde and then into acetate. Acetaldehyde is toxic. It binds to proteins and disrupts their structure. It also increases oxidative stress. So now inside you, there are two pressures at once: excess accumulation and toxic metabolite buildup. Your mitochondria are working harder, but their efficiency begins to drop. ATP production becomes less stable. You are still alive and functioning, but your internal environment is no longer perfectly balanced. Then comes inflammation. It does not arrive as a single event, it arrives as signals. Cytokines, small signaling proteins, reach you from immune cells in the liver and from circulating blood. Tumor necrosis, factor alpha, interleukins, and other mediators begin to alter your gene expression. Some of your hepatocyte neighbors become stressed and start releasing danger signals. Kupffer cells, the resident macrophages, become activated. They begin producing more inflammatory mediators. At this stage, you are not just a metabolic cell anymore. You are inside an inflammatory environment. The space of display around you becomes slightly altered. Stellate cells, which normally store vitamin A quietly, begin to respond to these inflammatory signals. They transform. This transformation is gradual but important. They shift from a calm storage state into an active contractile collagen producing state. Collagen is a structural protein that forms scar tissue. It is useful in injury repair, but harmful when excessive. As collagen begins to accumulate, the space around you slowly becomes less open. Exchange between blood and hepatocytes becomes slightly less effective. Nutrients still arrive, but diffusion is not as smooth. Oxygen still comes in, but gradients become altered. Inside you, the endoplasmic reticulum begins to feel stress. Protein folding becomes less efficient because of oxidative stress and altered metabolic demand. Misfolding proteins accumulate. You activate internal quality control systems to degrade them, but this adds more workload. Now consider a viral infection entering your world, such as hepatitis viruses. These viruses do not attack you from the outside in a dramatic way. They enter through blood, attach to receptors on hepatocytes, and enter your cytoplasm. Once inside, they hijack your ribosomes, which are your protein making machines. Instead of producing only your proteins, albumin, clotting factors, metabolic enzymes, you begin producing viral proteins. At first, you do not know anything is wrong, but your internal machinery is being redirected. Your endoplasmic reticulum becomes crowded with viral replication complexes. Your normal protein output decreases. Your stress increases. Eventually, your immune system notices. Cytotoxic T cells arrive. These are immune cells designed to kill infected cells. They recognize viral antigens presented on your surface. Antigens are small protein fragments displayed by infected cells to signal what is happening inside. Once T cells recognize you as infected, they release perforins and granzymes. Perforins create pores in your membranes. Granzymes enter and trigger apoptosis, which is programmed cell death. From your perspective as a hepatocyte, this is not a clean shutdown. It is a controlled dismantling. Your DNA is fragmented, your membranes are reorganized, your organelles are broken down in an orderly way. You are removed to protect the larger system. But you are not alone. Many hepatocytes are replaced continuously. Nearby cells divide to fill gaps. The architecture of the liver tries to preserve itself. However, if this process continues repeatedly, viral injury, inflammation, fat accumulation, toxin exposure, the environment around you changes permanently. Stellate cells keep depositing collagen, fibrous septa form, blood flow becomes distorted, oxygen delivery becomes uneven. Even some areas become underperfused while others are overloaded. You, as a hepatocyte, may now find yourself in a region where conditions are no longer optimal for survival. Nutrient exchange is reduced, waste clearance is impaired. Your ability to regenerate is still present, but the scaffolding that supports organized regeneration is disrupted. This is the beginning of cirrhosis at the cellular level. Not a single collapse, but a slow rewriting of your environment. Even then, you still attempt to function. You still try to process glucose, still try to detoxify blood, still try to produce proteins, but your outputs become less consistent. Your mitochondria become less efficient. Your internal stress responses are constantly active. And yet, even in this altered state, there is no dramatic moment where everything stops. There is only continuation under constraint. Still, blood arrives outside you. Still, signals pass through you. Still, you respond in whatever capacity remains, because even damaged, even stressed, even surrounded by scar tissue, you are still part of the system that keeps the organism alive. You are still the hepatocyte, but now your world is no longer stable in the way it once was. The environment around you has changed shape. It is still the liver, still organized in lobules somewhere in the larger structure, but the neat architecture you once relied on is becoming distorted. Fibrous tissue, collagen, laid down by activated stellate cells, has begun to form uneven bridges through what used to be open, flowing space. These bridges are not functional in the way normal liver tissue is. They are stiff, inelastic, and they bend the flow of blood away from its original efficient paths. Blood still arrives from the portal vein, but now it does not always reach you evenly. Some channels are narrowed, some are compressed, some are redirected. Flow becomes turbulent instead of smooth, where once there was a small uniform exposure of blood across hepatocyte plates, now there are areas of congestion and areas of deprivation. You are still receiving nutrients, but not with the same predictability. This change in flow has consequences that ripple into your existence. Oxygen delivery from the hepatic artery still continues, but diffusion becomes uneven because the microarchitecture around you is no longer symmetrical. Some neighboring hepatocytes begin to struggle slightly more than others. The system is no longer a perfectly cooperative grid, it is becoming patchy. Now, because blood is struggling to pass through the liver properly, pressure begins to build backward into the portal system. This is called portal hypertension. But from your perspective, it is not a named condition. It is simply increased force in the blood arriving around you. The portal vein, which normally delivers blood at a relatively low pressure suitable for slow exchange, is now pushing against resistance. You feel this as congestion in flow, not as sensation but as altered mechanical environment. The sinusoids become more engorged in some regions, stretched in others. Exchange becomes less efficient. Blood lingers too long in some spaces and too briefly in others. The careful balance of exposure time that once allowed you to precisely regulate metabolism is disrupted. In response to this chronic injury environment, regeneration attempts begin again and again. You or your neighboring hepatocytes attempt to divide. Cell division in the liver is not random. It is usually tightly regulated and organized so that architecture is restored correctly. But now the scaffold is distorted. Fibrous tissue acts like misplaced walls. So when regeneration occurs, it does not always reconstruct the original neat lobular design. Instead, nodules of regenerating hepatocytes form, small islands of attempted normality surrounded by scar tissue. These are regenerative nodules. From inside one hepatocyte, you would not see them as nodules, but you would experience their consequence: uneven distribution of workload, irregular blood supply, and inconsistent metabolic demands. Some hepatocytes are overloaded, processing more blood than they should. Others are relatively deprived. You are part of a system trying to rebuild itself while the blueprint is partially destroyed. Inside you, regeneration begins at the molecular level with signals. Growth factors arrive. Hepatocyte growth factor, or HGF, transforming growth factor signals in complex balance cytokines from immune cells. These signals tell you to re enter the cell cycle. Normally, a mature hepatocyte is quiet, stable, focused on function rather than division. But now you receive instructions to divide. So your internal machinery shifts. The nucleus begins expressing genes related to proliferation. Cyclins and cyclin dependent kinases activate. These are proteins that control progression through the cell cycle. You move from a resting state into a preparatory state, duplicating DNA, checking for errors, preparing for mitosis. This is not at all effortless. DNA replication is energy intensive. Mitochondria increase ATP production demands. Oxidative stress may rise further, but you proceed because the environment demands replacement. You split. From one hepatocyte, two daughter cells are formed. They are not identical in behavior immediately. They inherit the same genetic material, but their microenvironment influences their function. One may be slightly more exposed to blood flow, the other more protected. One may be more metabolically active, the other more focused on structural recovery. But this regeneration is happening in a distorted landscape. So while new cells are created, they are inserted into a framework that is already partially fibrotic, which means that even newly formed hepatocytes may not be perfectly aligned with the original sinusoidal architecture. At the same time, portal hypertension continues to affect the entire system. Because blood struggles to pass through the liver, it seeks alternative pathways. From the perspective of the whole organ, this means collateral vessels begin to form outside the liver. These are alternative routes for blood to bypass resistance. From your perspective, you may sense less controlled inflow, less organized exposure, and more irregular perfusion patterns. Meanwhile, immune activity continues. Kupffer cells remain present, but their behavior is altered by chronic inflammation. They are no longer simply cleaning occasional debris. They are continuously activated, releasing inflammatory mediators. This keeps stellate cells in an activated state, which maintains collagen production. A feedback loop is formed. Injury leads to inflammation. Inflammation activates fibrosis. Fibrosis distorts flow. Distorted flow causes more injury. Inside this loop, you are still trying to perform your core functions. You still receive glucose signals and attempt to maintain blood sugar balance. You still process amino acids and try to prevent ammonia accumulation through the urea cycle. You still detoxify substances using cytochrome P450 enzymes. You still produce albumin and clotting factors, but the efficiency of all these processes is no longer uniform across your structure. Some regions of you are still relatively functional, almost normal in performance. Other regions are under severe stress, with reduced oxygenation and impaired exchange. And yet, even now, there is no single moment where you stop. You continue adapting, you continue reorganizing, you continue dividing where possible and functioning where possible. Even in distortion, even in scarred architecture, even in uneven flow, you remain part of a system that is still trying to survive. Blood still arrives, though altered. You still respond, though strained. And the story does not end, it only changes shape. I hope you loved the narration of this. And I have realized that I have yawned a few times, but I mean, at least you know I'm not AI by now. And listening to yawn also helps other fall asleep, so I included it in the recording. I hope you don't mind. Good night.